Okay, so we're now at the last talk, and we're very delighted to have Hopi Hoekstra here today. And I have to say that really, who would you want other than her, given her work to expand and really give us such marvelous information about the workings of natural selection. So I think she's just about the perfect person. But she's been charged with a very broad task, which I'll mention her title in a second, and you'll wonder how she could possibly do it. So I know you're with the edge of your chairs for her whole talk. She got her bachelor's degree at UC Berkeley and got her PhD at the University of Washington. Then she had a postdoc in Arizona. Then she went to San Diego. That pissed me off because we offered her a job here. I'm sure a lot of other people did as well. She was there for a while, and then she went to Harvard, and I guess the non-proletarian Ivy League. And and she's now the John Loeb Professor in the OEB department, but also Curator of Mammals at MCZ. And her title is Evolutionary Biology, the Next 150 Years.
Thanks. Can everybody hear me okay? Yeah? Okay. Better? Okay. Okay. So I just want to be crystal clear from the beginning that giving this talk was not my idea. When Doug, I think it was Doug, sent me an email, and the idea was that Doug was going to give this, and did, this beautiful talk about the sort of last 150 years and all that we've learned, sort of being an established member of the field. And then what he told me was that we'd like to have sort of a young perspective. And I think I was so flattered by the fact that I was still considered young that I didn't stop to think that, in fact, maybe the strategy was that if they asked somebody who was more well-established, they wouldn't be naive enough to accept an invitation like this. Okay, and then to make matters worse, I talked to Mike Bell, and he sort of said, well, while you're predicting where the field's going to go in the next 150 years, why don't you just try to summarize the meeting as a whole as well? Okay, so I'm telling you this for a couple of reasons. To warn you that this is not the type of talk that I like to give, to generate some overall sense of pity, and also mostly to suggest that if this is a giant disaster, you know why. So I wanted to sort of preface my talk that when I started thinking about this, I realized what a challenge this is going to be. So please bear this in mind. So how I decided to approach this, as I do when there's a difficult question that I have to answer, I think it's always a good idea to ask this question. What would Darwin do? So if we ask what Darwin in 1859, can you predict what the field will look like in 150 years? Do you think he would have gotten it right? I'm not so sure. So let's think about it in this way. So let's think about some milestones. And I'm blatantly stealing this from David Kingsley, who pointed out to me that about every 50 years we seem to have a great milestone. So of course in 1859 there was the publication of The Origin. About 50 years later we had this rediscovery of Mendel's law. About 50 years after that we had the discovery of DNA. And then arguably one of the milestones of the current generation is our ability to sequence complete genomes. Would Darwin have gotten this right? I would argue that he might have gotten this 1909 discovery of Mendel. He was thinking about inheritance, right? I mean, he had his own theory. It was wrong, or wrong. That's okay. But he was thinking about this, so he may have said, "Well, we'll know something about inheritance in 50 years, right?" But I don't think he would have gotten the DNA part right, and I'm not sure he would get the whole genome sequencing part right. So, granted that we're all here celebrating what a prescient thinker Darwin was, I'm going to alter my talk based on what we just discussed, the talk title. And instead of talking about the next 150 years, I'm going to talk about what I think may happen in the next 1 to 50 years. Okay? Okay. So in an attempt to sort of think about, is there an overarching question that all of us here are interested in? So I've been sitting, listening to talks and taking notes furiously, trying to come up with some sort of common theme. And I think the best that I could do, at least in the last couple of days, if we want to sort of think about what's a big question that unites evolutionary biology. And I would argue at least one of them is this question about how variation is generated and maintained. I was struck by this because John Wakely, who's a theoretical population geneticist, said that his field is really motivated by answering this question. And then we saw Greg Ray come up and give a talk and had this beautiful slide of diversity saying, well, the field of Evo Devo is really interested in generating how this variation is generated. And then Rick Harrison ended his talk with a slide about diversity. And then we heard talks today about adaptive radiation, both current sort of adaptive radiation or contemporary patterns, and then patterns in the fossil record, interested in at least rates of how this variation is generated and maintained. So I would argue, or at least propose, that this is one question that we can all get interested in. And so today I'm going to try to guess or predict or outline some way where we can, in the next 50 to 150 years, try to answer this question in a broad way. So there are going to be several themes that I hope you can pick up on through this talk. So where are we going in the future? I would argue that I think future studies may certainly increase in scale. We are already seeing that happening with this genomics era. And I would also argue that this is going to be really enabled primarily by technological advances. So I'm going to mention a lot of potential technological advances, not because I think those are necessarily what drives the big questions in evolutionary biology, but I think a lot of the questions we want to answer are much more answerable when we have these sort of increases in scale, increases in our ability to gather data to answer those questions. You'll also hopefully see that, in the future, I predict that we get more and more integration among subfields. And that's not just subfields within evolutionary biology, but connections between evolutionary biologists and molecular biologists, evolutionary biologists and engineers and chemists and so forth. And then because I think that in the future we're not going to be limited by data acquisition, I think we're going to be much more limited by our ability to deal with all the data that we're going to be able to collect, I would suggest that future studies are really going to benefit from, if not require, collaborations. Okay. So if our grand challenge is really to understand evolutionary change, I think what we need to do is really get a sense of the complete picture of evolutionary change. That is, processes happening at the genotype level, how changes at the genotype level through development produce variation in phenotype, how variation in phenotype is then acted on by natural selection or other evolutionary forces in its interaction with the environment. So really what we need is to have this integrative picture. And of course, we want to do this through time. We want to know about changes in genotype through time, whether that's through comparative genomics or ancient DNA technologies. We want to know about changes in phenotype through time, whether that's using phylogenetic approaches and ancestral reconstruction or through the fossil record. And then we also want to know about environmental changes through time. But actually, one of the reasons I think this is a grand challenge is because it's really hard to imagine doing all of this, right? So if we take the simplest case scenario where just for one species and one trait, we want to understand the genes that control that trait, we want to learn about how that trait influences fitness in natural populations, it's actually a really hard thing to do, much less think about doing this for lots of traits, lots of organisms in a complex environment and know something about how that changes through time. Well, this is a grand challenge, but let me mention one thing. If even we want to do this for a single trait in a single species, this is going to require a lot of knowledge, right? So we have to know things about what's going on at the genotype level. So this is like the type of work that John Wakely and Andrew Kern talked about. We need expertise like that from Greg Ray, where we can understand how those changes through development affect phenotype. We need the expertise of organismal biologists like Hannah Coco and Jonathan Losos, and then we need expertise by ecologists like Mark McPeak. So really doing this is a big challenge. But this grand challenge is actually not a new one. I would argue that it was first sort of put forth in this type of way by Dick Lewington in his book in 1974, in the first chapter of that book, which was entitled The Problem, in which he suggested, even back then, that we knew a lot about what was going on in genotype space. We knew something about what was going on in phenotype space, but really the links between them were missing. Okay, so if Dick Lewington proposed this as a grand challenge almost 35 years ago, what have we been doing? Have we made any progress? And I think we've made a little bit of progress. So I can't give a talk without showing at least a few pictures of mice. So I think that, in fact, in the past, I don't know, 5 to 10 years or so, we actually have a handful of examples. This is not meant to be comprehensive. These are just a few of my favorite examples, where we actually know some genes that affect phenotype, which are important to the survival and reproduction of organisms in the wild. So, for example, we know something about genes that affect color that allow mice to be more camouflaged in their natural environments. We know something about genes involved in pigmentation or the loss of pigmentation in cavefish. We know something about armor plating and the genes affecting that in stickleback. But this is really let me pick on our own system for a moment. This is really the simplest case scenario. So in our work, we've started with pigmentation. Already we know that there's a general pattern that coat color of rodents match the environments they live in. We know this has an effect on fitness. We have a great list of candidate genes. Pigmentation's been studied for over a century by geneticists who study laboratory mice. And we know that only a few genes are involved in these traits. So here is the absolute simplest case scenario. So we've started to make progress, but only at a very simple level. So why am I so hopeful that in the next 50 years we can make any progress in doing this for a lot of organisms, for a lot of traits, and doing this through time? Well, one idea is that genomics is getting easier and easier. So I don't think I have to argue this point with this audience, but let me just highlight this by mentioning two things that have happened during the course of this meeting. Okay, so today's Saturday. Back on Thursday, this article was published in Science. That is a group in Mountain View. Were able to sequence the human genome for about $4,400. Okay, and they did 3 genomes, and this is the cost of reagents. Which is super exciting. The cost is falling incredibly dramatically. So if you can read this last line, do you have a pointer? I mean, this is Andy Kern mentioned this as well. So let's see. Just 6 years ago, the cost of sequencing a human genome was estimated at $300 million. In 2007, this was down 300-fold. It was just $1 million. Last year, it was $60,000, and now we're down to less than $5,000. So when Rick Harrison, you said that you didn't think your cricket was going to be sequenced anytime soon, but I bet you could afford to sequence your cricket on your own. Maybe. It's probably even cheaper because the genome might be smaller than humans. Okay, and then yesterday in Science, there was another article, and this was an article written by Liz Panisi announcing the sort of official plan of the 10,000 Genome Project. The idea is that 10,000 genomes of vertebrates would be sequenced. So this is about 1/6 of all known vertebrates, the idea being that they were going to sequence one from every genus. Not only are genomes being sequenced sort of broadly, but of course singles. We're also sequencing genomes, many genomes within a single species. So many of you are probably familiar with this 1,000 Genomes Project, the idea that we're going to sequence the complete genome of 1,000 humans. Of course, this is more biomedically related, but Drosophila geneticists are never far behind. And so the next 1,000 Genome Project has been proposed by Drosophila geneticists, the idea here being that they would sequence 600 melanogaster genomes to do the type of population genomics that Andrew Kern was talking about, but then of course also 300 more so that they could look at temporal versus tropical populations to get at the question of local adaptation, and then 100 more looking at sister species comparisons. Okay. So there's a lot going on in the field of genomics, and it's not just sequencing genomes. It's sequencing, as Craig Venter has been promoting, a scoop of soil or a scoop of the ocean to try to understand biodiversity, a field called metagenomics. There's, of course, transcriptomics, proteomics, metabolomics, epigenomics, ionomics, physiomics, and I'm sure you guys could add more to this list. Okay, so there's no doubt we're going to be, we are and will continue to be swimming in data. I think what's going to happen, or I hope is going to happen in the next few years, is we're going to increase our ability to deal with all this data. How are we going to do that? Well, hopefully with the help of some bioinformatics folks. So there are lots of genomes that are being sequenced, but how are we going to annotate all those genomes? Where are we going to put them in terms of a repository? How are we going to make those genomes accessible to all of us who aren't, excuse me if I offend anybody, most of us maybe who aren't bioinformaticians, and then how are we going to have the computing power? So I just mentioned that 10,000 vertebrate genome project. They proposed to sequence all of these for less than $50 million in the course of 5 years. If that's true, what that means is once every day we'd have to assemble and annotate a genome. Okay? So this is going to be a really important part of the equation. Okay. I'd like to argue the point that genomics isn't going to solve this question on its own. So the question is, what can genomic data tell us about the evolutionary process? What can we learn from genome sequences alone? I think we can learn a lot, and that's been obvious, and we've heard a lot about that in this conference. But I think there's some questions that we can't learn. So let me just ask this sort of in a silly kind of way. If this is an important question to you, will we be able to, how an elephant's trunk evolves, can we do that just by sequencing the genome of an elephant? And I think we can all agree that we can't, and this seems obvious to us now, but I think that if we look back even just a few decades ago, that wasn't so obvious. So let me pick on Sidney Brenner for a minute. This is a smart guy, right? This is a guy who was involved with Francis Crick in proposing the idea of messenger RNA. He was involved in showing the experiments that showed that the genome code was triplicate in nature. Not to mention he won a Nobel Prize for promoting C. elegans as a model in development. Okay. Maybe we can attribute this to sort of blind optimism, but he was known, I don't have the exact quote, so bear that in mind, in a talk given to at Cambridge University on the occasion of Darwin's 100th anniversary of his death, so this was in 1982, to say something to the effect of, give me the complete DNA sequence of any organism and I can reconstruct that organism. Seems kind of silly to us now, but at the time I think there was this optimism that in fact if we had the genome sequence we could solve everything. And unfortunately I think that's not true. So where I hope the field is going after we start to deal with all this genome data is to try to understand how these genes interact with each other to produce phenotype. Okay, here this giant mess is just protein interactions from a simple bacteria. So now imagine scale that up to something as humans. This is going to be no small task, sort of understanding how a change in the genetic level produces a change in phenotype level. But that's the one benefit of giving this talk is I don't have to tell you how we're going to do it. I can only tell you that I think we're going to do it. And so what I think is going to happen is that, or what I hope is going to happen is that we're going to start to use high throughput techniques to understand expression patterns. And then, of course, we need some functional genomic tools, hopefully high throughput technology to knock out genes, to express genes in wrong places. And that hopefully these tools are easily transferable to other organisms. Because you may argue that this type of work, we should leave it up to the molecular biologists because they can knock out every gene in the Drosophila genome and see what the phenotypic effects are. But I think that evolutionary biologists have a lot to lend to this type of work because we're interested not in all the genes that are necessary to produce a leg or a hand. We're interested in the specific gene changes that make a hand that looks like this go to a hand that looks like this, right? So we're interested in changes in this network. To make the connection between genotype and phenotype, not only are we going to have to sort of understand this, But I think we also have to do a better job of measuring phenotype. Okay? So while our depth of knowledge for understanding genome biology has been increasing rapidly, I would say that phenotype biology, organismal level biology, hasn't been keeping up in the same type of way. Right? There's still many of us, I included, who go to the field, measure phenotype with a pencil in our little notebook. We measure mouse tail length, their foot length, etc. This is a paper that came out last week in PNAS where David Houle makes the similar type of argument. If we're really going to make the link between genotype and phenotype, not only do we have to get more efficient and more consistent in measuring phenotype, that is increasing our ability to measure many traits, or to measure a particular trait in many individuals, we have to be smart about what phenotypes we choose. So again, we can turn to this HapMap project, and there was a lot of enthusiasm that the HapMap project was going to allow us to identify every disease allele in the human genome, and that largely hasn't borne true. I think we've learned a lot, but I would say it's not as successful as we thought it might be. I don't know if that's a politically safe enough argument, but one argument for why that has failed, has not been as successful as it might have been. Sorry, this is not being recorded, is it? Is that we haven't been really good at measuring phenotype. So there's lots of problems. One of them, arguably, is this rare allele effect. So there are multiple alleles that can affect a particular disease, but that's exacerbated by the fact that, for example, if we're looking at the genetic basis of a complex disease like schizophrenia, and we're comparing people who are diagnosed with schizophrenia versus another pool that is considered healthy, and we want to associate genotype with phenotype, the question is, how do we know that these are all considered, these individuals are all disease individuals or all schizophrenic, if different doctors are diagnosing them using different traits? And probably what we're doing is lumping a lot of different subtle diseases into a gross category called schizophrenia. If we're not measuring, the point is, if we're not measuring phenotype right, then our hope of making that association between genotype and phenotype goes down dramatically. So we have to get better at measuring phenotypes. I think we're also already making some progress in this. So I stole this picture. This is a snapshot of Morph Browser by Junke Jurval's lab, where they're interested in the evolution and development of teeth. And they've devised this way of systematically measuring tooth morphology. And this sort of phenomics approach can be used at lots of different levels, macroevolutionary levels when we're looking at fossils, if you're interested in natural populations, or if you're looking at mouse mutants. So that's a standardized way to measure lots of aspects of tooth morphology in a consistent and large way. Okay, this is just one trait. Let me just say that there are actually companies that do this now. This is largely motivated by either disease, biomedical interest, or agricultural interest, but this is the type of thing I think evolutionary biologists can take advantage of too, because they're doing high throughput screens of, let's say, plant phenotypes, where they're measuring absolutely every aspect of this plant, from physiology to behavior in terms of plant behavior, moving towards light, etc. Everything in this room. These mice that get subjected to these tests, there's like 300 different tests that these mice are subjected to. So we're doing a much better job at being able to measure phenotype. Okay, but as we know, phenotype in the lab is different than phenotype in the wild. So if we really extend our imaginations, what we'd like to do are these types of tests where we measure phenotype, but we do it in an environmentally relevant way. So wouldn't it be great if we could just tag, let's say, hundreds of individuals of your favorite species and watch what they're doing, when they die, when they reproduce, where they move, what their local environment is? Well, in fact, this work is also started. In collaboration with engineers, biologists are devising little monitors here, which is some sort of cricket. You can see this little instrument here. These are being miniaturized. Here is on the back of this moth. We can start to think about this connection between phenotype and environment in a realistic way. But maybe it's not enough just to track movements of animals. Well, just let's step back for a second. Monitoring all these might be hard, but maybe that's not as hard as we think. So there's a new project called Icarus that's actually co-opting satellite power to actually do this all in an automated way. So what if you could measure your individuals, where you knew something about their genotypes, you're interested in their phenotypes, and then you could put them out in the wild and start to measure what they were doing without even leaving your office? Pretty cool. And maybe it's not even just that we want to monitor their movements, but what if we could also use little tools that measure their physiology, how warm they are, how much energy they're expending. Just think about all this data that could be possible. We'd also like to monitor the environment. Just to mention, there's some projects that are, again, already starting to do this. This is the National Ecological Observatory Network. It's a program funded by NSF, where there's a global effort, or an effort to monitor environmental change globally. As I mentioned, this is an overwhelming amount of data. And so I think this is going to require large-scale collaborations, integrations across genome, phenotype, and environment. This is just an example of such a collaboration called iPlant, which is meant to bring together people with an interest largely anchored by genomics, so this plant genomes, and provide a cyber infrastructure for collaborations. Okay. So what I think is going to happen, what I hope is going to happen, what I will predict, you can check in 150 years, what will happen is we'll have a much more integrative picture of evolutionary change. But as I mentioned, I was just going to talk about the next 50 years, and I think that, or I hope that, we're going to see a lot of effort being put at making this connection between genotype and phenotype and resolving this genotype-phenotype map. And let me tell you about why I think that's exciting in just a second. Now, of course, we can't do this in a single organism or for a single trait. So let me just put in a plug to mention that I think we're going to get a much bigger picture as we're starting to develop what I like to refer to as emerging model systems. So this is already underway, and I think that there are a number of other model systems, even Rick's crickets, that may, in fact, tell us a lot about evolutionary process. As I mentioned, we're very far along for an inordinate number of species. So here we're just looking, this is a plant clade, this is an animal clade. These circles represent different genetic and genomic tools. And when they're filled, that means those resources are already available. So here's natural variation, a genetic map, our ability to do experiments, to culture them, to do transgenics, reverse genetics, forward genetics, EST library, back library, or whole genome sequence. Look at how many of these circles are already filled in. This can only get better. So I think there is a real possibility that we can start to answer these questions across systems. Okay, so what if in 50 years we have a genotype and phenotype map? What can that tell us about the evolutionary process? Okay, so probably the first step we'll make is making this connection between genomes and morphology, because morphologies are inherently relatively speaking, easy to measure, especially if we get those phenomic tools. And we have some understanding about how development translates these two. If we're able to find genes, then we can start answering these questions that are sort of long-standing questions that date back almost 1,000 years, 100 years. So for example, this is a question that predates the field of population genetics, that really was the argument between the biometricians and naturalists. So how many and what are the effect sizes of genes that contribute to adaptive phenotypes? Do adaptive alleles tend to be dominant or recessive? This was a topic of debate by J.B.S. Haldane. What is the relative role of epistasis versus additivity? Wright really argued that epistasis was an important component to phenotypic change. Where do alleles come from? Are they pre-existing or are they mutation limited? Are the same genes responsible for convergent phenotypes? This goes back to that question of constraint that Mark Kirkpatrick and Gunther talked about. Are adaptive mutations protein coding or cis-regulatory? Okay, this is just an example from of what finding the genes may help us understand about the evolutionary process. Okay, but not all of us maybe would consider themselves evolutionary geneticists. What if we knew something about the phenotype-genotype map? Could we lend something to the study of paleontology? Well, what if we could get some ancient DNA, sequence a few genes? Could we predict the phenotypes of extinct taxa? Okay, and David Hillis, this is for you because you said every talk so far had a tree in it, and I wanted to make sure that I didn't ruin your trend there. So we could also, instead of having ancient DNA, we could potentially reconstruct some ancestral sequences. David already gave us an example of this. Wouldn't that be cool if we could, just based on genomic data, start to reconstruct what phenotypes look like for organisms that don't exist anymore? And in fact, I'd argue we already can do this, at least in one case. So let me tell you very quickly that in these mice, we know that a gene called the melanocortin-1 receptor has a single amino acid that turns a dark mouse into a light mouse. Well, some colleagues in Leipzig sequenced the same gene using ancient DNA techniques in mammoths. They found not only a mutation in the melanocortin-1 receptor, they found the exact same mutation, an arginine to cysteine change at position 65, which suggests that mammoths, like these mice, may have been polymorphic in color. So this is just one simple example from one simple gene that maybe we can start to reconstruct phenotypes. And maybe Tim isn't satisfied with this because it's not humans, but let me just say that we could maybe start to do this for hominids and primates as well. It turns out that the same melanocortin-1 receptor is also involved in human hair coloration. Alleles that are high frequency, for example, in Irish populations, result in red hair. Well, we tend to reconstruct Neanderthals like this, sort of a dark, swarthy look. Well, we sequence this, not we, folks sequenced this gene in, again, a group from Leipzig, in Neanderthals using ancient DNA techniques, and found that this melanocortin receptor, when subjected to functional assays, behaved just like the red hair receptor in humans, suggesting that at least some proportion of Neanderthals also had red hair. Super cool. I'm going to skip this, but just to say that I don't think it's only a unidirectional transfer of information from genomes to phenotype. I think that we can actually learn from that phenotypic information can teach us about developmental genetics. I think the work by Neil Shubin actually highlights this quite well, that by targeting fossils in doing sort of, what does he call it, targeted expeditionary whatever events, he can find these fossils, and that can actually tell us about or generate predictions about what we may expect at the genetic, or in this case, developmental level. Okay, what about Hanakoko? What about behavior? These same exact tools and techniques that we can use to connect genome or genetic data to morphology can be used to identify genes underlying behavior. We have a good example now of the vasopressin-1 receptor affecting social behavior in voles. Those of you interested in speciation, whether that's directly in speciation like Rick, or those interested in adaptive radiation, or at a macroevolutionary scale, the rate of origination, I think that we can also treat reproductive isolation as a trait and try to identify genes that contribute to barriers to gene exchange. And this can, again, inform our understanding of the evolutionary process. Okay. So if I had to predict, and we can argue about whether these are good predictions or not, and you can make your own maybe over a glass of wine later, but if we start with 2009 and our whole genome sequencing ability, where will we be in 50 years? My hope is that we'll at least have a more complete idea, a more complete picture of this genotype to phenotype map. Because wouldn't it be cool if we can predict, based on DNA base pair changes, what the effect would be on phenotype? I hope to have given you some examples of that. Maybe give us another 50 years. Maybe we can start to understand better how these changes at the genetic level affect phenotype and how those phenotypes affect fitness in nature. I put this back 50 years because I think this is an inherently more difficult problem, right? There's much more dimensionality to the natural environment than there is to the genome. And then maybe, this is in part to try to say something about paleontology, to pull that into the talk, in 150 years, we can start to reconstruct ancestral phenotypes in ancestral environments. Okay, let me end by just acknowledging the following groups. So I've been fortunate enough to be involved in an NSF workshop called the 21st Century Biology that really brought to my attention some of these cool technological advances that I actually don't think are that far away. As well as an nascent working group on emerging model systems. And I had some very exciting and animated discussions with Dick Lewinson about this, who also has strong opinions. And with that, I'm happy to address any questions. Thanks.